Hello everyone, am I audible?
Yes, ma'am.
My slides are visible?
Yes, ma'am.
Okay. So let's start the session. So this is the module 3. Up till now, we have studied the module 1 and 2, in which we have studied about the insights of what a gut microbiome is made up of, how many different type of bacterial groups are seen, and also their overall role in boosting our immunity, in preventing us from so many diseases, and also in protecting us from mental disorder, like the role of gut microbiota in controlling different kind of neurological disorders. So we have studied the very basic thing and up to some in depth also, but the module 3 is intermediate to advanced level. We will see everything in a bit much more detail. So the first topic is gut microbiota in gastrointestinal disorders. So as you all know, the gut microbiota has a fixed composition, that A, B, C, D bacterial group will be there. And if this composition is somehow altered due to several factors, and we have already studied the factors responsible for the dysbiosis. Dysbiosis is imbalanced gut microbiota. So if the composition of good amount of bacteria or good bacteria is somehow altered, then it may lead to different kind of diseases, not only just gastrointestinal, but also different metabolic disease like diabetes, obesity, and it has also several complications related to the central nervous system. So as you all know, whenever a natural thing is somehow altered by us, so it may lead to different kind of disorder and disease. So all these things are very important, and we will study this first part. We will start from the gut microbiota in gastrointestinal disorder, like how these good bacteria may lead to gastrointestinal diseases. What are the different kind of gastrointestinal disorders? So specifically, we have to study about the IBS, IBD, and celiac disease. So we will focus it one by one. Firstly, we will study the celiac disease. Then we will study the IBD, IBS. So let's start. So whatever I was just discussing with all of you, that gut microbiota is a highly dense population of different kinds of bacteria, A, B, C, and D, residing in our gut, which again co-evolves because they show a symbiotic relationship. They show a mutualistic relationship with the host. They benefit it in every possible manner. So all these gut microorganisms are involved in different kind of metabolic as well as immunological activities. So this gut microbiota can be considered as our very important part of our body. If we want to run our whole metabolic machinery very smoothly and without any disease or disorder or any distress, so we can say that we need to have a good gut microbiome. If our gut microbiota is altered by any factor, then it may lead to different disorders and disease, not only just physical disease, but also mental disease. So gut microbiota is associated with maintenance of health, and unbalanced microbiota contributes in the development of several diseases. If you are the one who is spoiling his or her own gut microbiota, then it will lead to different disease. So alteration of beneficial gut microbiota population triggers gastrointestinal diseases, including irritable bowel syndrome, inflammatory bowel disease like IBS, IBD, celiac disease, and the most important one nowadays is colorectal cancer, because so many people are nowadays diagnosed with this colorectal cancer, and they are having very common symptoms which we mislead that this is normal indigestion or some diarrhea thing, dysentery thing, but that may lead to several severe disease like colorectal cancer. So the major disease which are IBS, IBD, celiac disease, and colorectal cancer. So all these diseases are related to the disturbance in the beneficial gut microbiota, which will result in different gastrointestinal diseases. So today we will focus on gastrointestinal diseases, but from tomorrow onwards or the day after tomorrow, we will study this also, that how our gut microbiota will influence our different metabolic machinery and may lead to metabolic disorders like diabetes, obesity, and NAFLD is non-alcoholic fatty liver disease. Because many a times we mislead that only uh, liver disease can be seen if we are consuming alcohol, but no, apart from alcohol, our food diet or our how different type of nutrient we are providing in our body also influences the liver. So this is known as the non-alcoholic fatty liver disease. So we will study this, and in the third part we will see the impact on autoimmune disease and the central nervous system, and the fourth one we will see cancer and microbiome emerging inside, like how these normal microorganism will lead to very severe disease like cancer. What are the different factors responsible for turning the normal gut microbiome into cancerous one? So the gut microbiota contributes to the differentiation and maturation of resident intestinal immune cells, including Th17 cells, regulatory T cells, innate lymphoid cell, and IgA producing B cell. So I have listed all these things in my previous classes also, that our gut microbiota or gut microbiome do what? It activates the T regulatory cell, the Th17 cells. So all these things are very important because they are the mediators of immunological response. So our microbiota is responsible, or you can say that all these cells are activated and stimulated by the microorganism residing in our gut. So this T helper cell like 17 cell, regulatory T cell, and innate lymphoid cell like B cell, and antibody producing B cell, plasma cells. So all these are activated to show different response whenever there is an encountered antigen in the body. So whenever we are encountering any kind of antigen, then all these things will be activated in a different manner. All everything has a definite cascade, so they activate and they neutralize the effect of antigen. So all these things will take place only when your gut microbiota are healthy, and a good there is a diversity and also abundance of good bacteria, so then only these things will be activated. But if you are having a leaky gut, if you are having dysbiosis, then these things will not be activated, and then what will happen? This antigen will be able to cause infection in your body. So we need to maintain a good gut composition. Now, normal role of gut microbiota. So as we all know, normal role of gut microbiota is to do what? Maintain intestinal homeostasis through different composition of bacteria. They are responsible for synthesizing short chain fatty acid, acetate, propionate, butyrate. We have studied about this butyrate, that what it do. It increases the uh, beta oxidation by the mitochondria. So ultimately it will do what? It will drain all the oxygen so that anaerobic conditions are maintained, because we are having obligate anaerobes. Obligates anaerobes are there. These are the good one, good bacteria in our gut. So if oxygen is not present, then they will increase their population, and if they will increase their population, they will synthesize butyrate, which is the mediator of all the metabolic chains. So they also aid in our digestion and absorption. They regulate our immune system, especially the gut associated lymphoid tissue, gut we have studied, and they prevent colonization by pathogens. So they also provide the colonization resistance. It means they prevent the increase in, or they inhibit the pathogen population to grow in a larger amount, because if they will grow, they will cause different kind of infection in our body. So it is the function, or it is the role of our gut microorganism to prevent the growth of pathogen in our body, so as to make us remain healthy. So all these things are very normal role of gut microbiota. Now, before this, if dysbiosis occurs, like if imbalance in gut microbial composition, it may lead to several gastrointestinal tract disorders or diseases. So the common one is IBD, irritable bowel syndrome or disease, sorry, and IBS, colorectal cancer, celiac disease, Clostridium difficile infection, CDI, and this one NAFLD, and gastric ulcers and gastritis. So everything is different from each other in terms of their etiology, in terms of their symptoms. Many symptoms are common for all of the disease, but it depends on the severity level, how much severity of a particular symptom in each of the disease is there. So every disease you have to study about that, what are the different kind of complications associated with this particular IBS, IBD. So first one is IBD. IBD can be IBD and IBS. You can compare. IBS is just a syndrome. This is not a severe thing as compared to the IBD. IBD is a severe form. We will compare it with a difference between thing, so you will be able to understand it in a clear manner. So in IBD, whose population is increased or decreased, so firmicutes, which are the good one, its population is decreased. Now the proteobacteria are increased, which triggers inflammation via toll-like receptors and anilars. So everything is done by the increase in the proteobacteria. If a bad bacteria is increasing, then what it will do? It will do different kind of inflammatory responses in our body. So if our body is having an inflammatory response, then our body is always in an always feel lethargic, because all the energy are drained in providing inflammatory responses. Now, apart from that, IBS, we will study this in a very detail. This is a just a chart that what are the different kind of diseases associated with the dysbiosis. If your gut is imbalanced, if you don't have a good composition of good bacteria, then what are the different kind of things can happen to you? So IBS, altered microbial metabolites, bloating, pain, and role of methanobrevibacter. Colorectal cancer, Fusobacterium nucleatum will increase, and also different kind of toxins they will produce to initiate the inflammatory responses. So every bacteria or every pathogen functions through secretion of some kind of peptides or any toxin. So everything is initiated when they release their product in the host. Now, celiac disease. Celiac disease is dysbiosis contribute to the loss of tolerance to gluten, which is the most common one found in wheat, rye, and likewise. So you can see so many people are allergic to gluten. So because of celiac disease, we will see this in detail. And CDI. CDI is Clostridium difficile infection. It is post-antibiotic dysbiosis, loss of colonization resistance. Like this is observed when you are for a very long time you are taking any antibiotic. So your normal gut microbiome composition will be somehow altered, and then what will happen? It will increase the population of pathogens. If pathogen population is increased, what will happen? If pathogen population will increase, they will release some neurotoxin or genotoxins or different kind of their toxin molecules in different biochemical nature. So it will lead to inflammatory responses. Now NAFLD is also a gut liver axis involvement, increase endotoxin from microbiota. So if you are not taking alcohol, still your liver is not working properly, still your liver profile test is not so good or appropriate, then you can have a impaired gut liver axis. Like your good amount of bacteria are very low and bad amount of bacteria are high in numbers. So there is an imbalance which is known as the dysbiosis. So dysbiosis can lead to what? It can also lead to non-alcoholic fatty liver disease. Now, apart from this, gastric ulcers and gastritis. Uh, when there is an increase in the growth of Helicobacter pylori, then there will be chronic inflammation. Your gut will feel always some kind of acidity. No food will be digested properly. Without eating anything, you will feel your stomach will feel always in a acidic condition. You will feel bloated, and this is because of the inflammation in the gut epithelial layer cell. The IEC, intestinal epithelial cell, will be inflamed. So this condition is known as gastritis. So all these things you can see whenever you are having a gut dysbiosis. So mechanism of disease induction, like the gut microbiome how the gut microbiome can initiate different kind of diseases, like the metabolic one. So the metabolic, if how they can influence the person's immune system, or to cause any kind of gastrointestinal disease or any disease. So basically what they do is, the first one or the foremost is metabolic alteration. Like if the gut microbiome composition is altered, then what will happen? They are known to synthesize short chain fatty acids. So if short chain fatty acids are altered, if they are not synthesizing the propionate, acetate, butyrate, instead of that they are synthesizing something else. So what it will do? It will not initiate the rest of the signaling cascade, because we know that butyrate is activating different molecules, and which is ultimately stimulating the beta oxidation of fats to reduce the presence of oxygen, to stimulate the growth of whom? To stimulate the growth of obligate anaerobes, which are the good bacteria in our gut. So first and the foremost one is metabolic alteration. The, the gut microbacteria will do what? It will alter the overall short chain fatty acid production. So short chain fatty acid production will, if it is impaired, then it will lead to overall uh, disturbance in your overall metabolic machinery. Now, how immunologically they can alter our different function. So modulation of T regulatory cells, T helper cells, and release of pro-inflammatory cytokines, that is interleukin 6, TNF alpha. So they may release different kind of inflammatory molecules directly into the bloodstream. If they are releasing it directly into the bloodstream, then what will happen? Our body will start producing the anti-inflammatory response. So all these things are done immunologically by releasing different inflammatory cytokines. Now, barrier dysfunction. Barrier dysfunction means what? Barrier dysfunction means we know that in our intact gut, one cell is tightly held with the another cell through tight junction. Nothing is leaky. But when there is an alteration or the rupture of this tight junction, then what will happen? Everything will be leaked out from the cell, and from cell to into the bloodstream. So this is the barrier dysfunction. Barrier dysfunction uh, is known as the leaky gut. So there are always these terms will be used: leaky gut, intact gut. So you should be able to recall the exact meaning of leaky gut. The whole picture should be in your mind while I am saying leaky gut, tight junction, or the dysbiosis. Everything should be a clear cut picture in your mind should be there. Now, fourth one is bacterial toxins. Some of the bacteria or pathogen are known to release different kind of toxins whenever they enter or invade the host immunological barriers. Like we are having firstly the innate physiological barrier. Skin is there, physical barrier. We are having physiological barriers like secretion, saliva secretion, tear secretion, acid secretion in our stomach. All these are physiological things. So when the pathogen is capable of invading all these barriers like physical barrier, chemical barrier, or physiological barrier, then they are capable of synthesizing different kind of toxins, and these toxins may alter the overall functioning or the normal functioning of the host body. Because suppose this is a normal body of a host. This is a normal body of a host, and this man is fit and fine when there is an antigen. This is a bacteria. When it will enter his body, so what will happen? His skin will try to resist this bacteria. First of all, our physical barrier, or in our innate immunity, we are having a first line of defense, that is our skin. Our skin will always try its best to warn, not letting the bacteria to invade our skin. So if our skin fails to do that, if this man's skin will be fail to prevent this bacteria to enter into his body, then what will happen? This bacteria will take entry into his body, and then what will happen? This has already passed the first line of defense. Now it will start synthesizing different toxins. And if different toxins are synthesized, what will happen? Suppose these toxins are lipopolysaccharide. So lipopolysaccharide will be overall in the blood. So blood flows from top to bottom, not only just in our heart, not just only in our liver. Overall body have the circulated blood. So all those blood will be having this toxin. So you don't know, your whole body is having more than trillions of cells. So a cell is like this. It has so many receptors. So you don't know when a molecule or any toxin can act as a uh, uh, any kind of stimulant. So if it will bind, it may replicate the other molecule. So what it will do, competitive inhibition may occur. So this will bind to the normal cell, and then it will initiate the inflammatory responses. So whenever there is a toxin in our bloodstream, it may lead to different inflammatory responses. So these four are the major contributors of disease whenever there is a uh, imbalance in our gut microorganism, or in short you can say dysbiosis. So you should write it down properly, the whole story in just 4 words, that how your uh, body shows different kind of diseases when you are having an impaired gut microbiome. So through these 4 things they show different disease. Like metabolic disease, how they show? Through altered short chain fatty acid production, altered bile acid metabolism. They will not show a normal metabolic pathway to do all those digestion of the important nutrient in our body. So they will not be absorbed in our body, and no energy will be there. And immune, how the immune system influences our body if we are having a imbalanced gut microbiota. So it do through the modulation of this T regulatory cell, T helper cell, and release of the important inflammatory cytokines like interleukin 6 and TNF alpha. The third one is barrier dysfunction. Barrier dysfunction, as the name suggests, the disruption of the tight junction. If the tight junction is not working properly, no cells will be in the close proximity, and they will show some leaky inflammatory molecule will be leaked out directly into the bloodstream. Now, fourth one is bacterial toxin. So, bacterial toxins are the toxin which are capable of initiating inflammatory responses. So, when there is a toxin production, it means it has already passed your first line of defense and it is showing the inflammatory responses in our host cell. So these are the four factors or the four mechanisms through which we can expect disease in our body if we are having dysbiosis. Now this table I already mentioned. Now this question, dysbiosis in inflammatory bowel disease is characterized by. Dysbiosis in inflammatory bowel disease is characterized by this B option. Increase in firmicutes and increase in proteobacteria. We have studied here that in IBD, what happens? The firmicutes population is decreased and proteobacteria population is increased. So, and dysbiosis, which will lead to what? Inflammatory bowel disease will lead to or is characterized by a decrease in firmicutes and increase in proteobacteria. So right answer is decrease in firmicutes and increase in proteobacteria. So you have to remember this hallmark that whenever there is a reduction in the population of this firmicutes, you can say that there is an imbalance going on in your normal gut microbiome composition. So remember this key point: whenever there is a disturbance in the normal firmicutes composition in your body, there might be surety that something is going on in your body, that your normal gut microbiota is altered or the good bacteria amount is decreased. Then these four things you can expect, like altered SCFA production, the release of pro-inflammatory cytokines, barrier dysfunction you can expect, and the release of toxins by the pathogen. So everything can be take place whenever you are having this thing, dysbiosis, the imbalance of the normal gut microbiome composition. Your normal microorganism is in low amount and you are having an increased population of what? An increased population of the bad bacteria. Now this is diagrammatically I can explain to you that environmental and immunological factors which will lead to dysbiosis, and ultimately this dysbiosis will lead to what? Gastrointestinal diseases. Like you can see the pathogenic role of the gut microbiota in gastrointestinal disease. Various environmental and immunological factor causes gut dysbiosis. We all know about those factors, like the environmental factor, if you are living in a pollution environment, the age factor. With age, the gut microbiome composition is reduced or the amount of good bacteria is automatically reduced. If you are having any kind of disease, any metabolic disease, whether you take alcohol or not, so all these are the major contributors of dysbiosis. So you can say environmental and immunological factors like alcohol, smoking, yes, diet I forgot here. Diet is the major contributor. Diet, antibiotics, aging, genetics, and stress. So whenever there is this kind of factors associated with you, then there might be chances that it can lead to reduction in the population of whom? The good bacteria. I have already told you that we know dysbiosis, the level of dysbiosis 1, 2, and 3. So we have read that the diversity is decreased in the level 3, like the good population bacteria also decreases and bad bacteria also decreases. So this is known as the diversity and richness will be decreased, like no different bacteria will be available in your gut, and the population will also be low, like only 10 bacteria will be there of each species or each variety. So you can say that the diversity and richness decreases whenever there is an environmental and immunological factors contributing to dysbiosis. So this is the first thing, that diversity and richness will be decreased. What will increase? Pathobiont. It means the microbiome or the microbiota, or you can say the bad bacteria, the pathogens, the increase in the you can see the increase in the population of the pathogens as the increase in the different kind of factors responsible for dysbiosis will increase. It will lead to increase in the pathogen. Now, immunogenicity. Immunogenicity means the immunological response by the gut bacteria or the gut microbiota will also increase, because the altered gut or the dysbiosis will release what? The inflammatory mediators. If the inflammatory mediators will increase, then it will lead to immunological responses, and that is known as the immunogenicity. Your own cell will be it can be considered as non-self, and whenever your immunity considered your own cell as non-self, then it will initiate different kind of immunological responses. Now I have already discussed about this point. Colonization resistance will decrease. Colonization resistance will decrease. It means the increase in the population of bad bacteria, that is the pathogens. Now what it will do? It will lead to abnormal immune response and malmetabolism, which will lead to the leaky gut, and the leaky gut is associated with different kind of gastrointestinal diseases. And also you can see here epithelial integrity will be decreased, because one cell is attached to the second cell by means of the tight junction. If tight junction is not functioning properly, then what will happen? It will lead to leaky gut. So again, this is an important point, that whenever there is an any kind of dysbiosis, and dysbiosis can be due to environmental factor and immunological factor, and out of all these two, alcohol, smoking, diet, antibiotics, age factor is also there, and genetic factor is also there. Somebody inherit the faulty gut microbiome composition from their parents, so this is the genetic thing. If you are a person who always take stress, depression, anxiety, frustration, so your gut microbiome population will be something different. So everything will be altered whenever there is an immunological or environmental uh, stress kind of thing. So if it will happen, if dysbiosis will happen, it will lead to leaky gut. Everything will be directly bath into your bloodstream. What will directly bath into your bloodstream? The inflammatory molecules. And what it will do, you can see here the diversity and richness will be decreased. Of what? Diversity and richness of what? Microorganisms. The pathobiont will be increased, like the pathogen will increase. Symbionts or the mutualistic microorganism will decrease. Immunogenicity will increase. Colonization resistance will decrease, and it will lead to decreased epithelial integrity. It will no longer be intact, and mucosal immunity will be decreased. Gut will be hindered or malfunction. Gut associated lymphoid tissue. So it will lead to different gastrointestinal diseases like IBD, IBS, celiac disease, colorectal cancer, and a lot more. Now we will start studying about the first disease in detail. This was all about the introduction kind of thing. So in our module 3, we are having 3 different diseases in detail. That is IBS, IBD, and celiac disease. So we will start studying it one by one. Celiac disease firstly we will see. So you can see celiac disease. So there are so many different kind of disease. You can just try to read the symptoms of different disease. You will see that majorly for many of the disease, many of the gastrointestinal disease, the symptoms are very common. You will not be able to distinguish properly in between IBS, IBD. The only thing is that which differentiate from each other is the level of severity. So everything depends on the infection level, severity, how much it can alter the the host immunity. So you need to study all these disease in details. So the first one is celiac disease, which is a chronic immune mediated inflammatory disease of the small intestine, is an autoimmune disorder triggered by the consumption of dietary gluten. So uh, why this is known as the uh, just a minute. Uh, why this is happening? The cursor is not functioning. Wait for a while. My laptop is not working. Oh no. Mouse is not working and touchpad is also hanged.